Very good morning to the students of class 12 science. Welcome to biology class. In last class we have learned about the cloning vectors and the characteristics, right? So today we will learn about the vectors which are being used for cloning genes in plants and animal cells. Clear? So mainly vectors are being used to transfer the foreign DNA or the gene of interest into the host, right? So we have learned about the process of formation of recombinant DNA technology also, and after their formation they are being introduced into the E. coli cell or the host cells in order to produce multiple copies, right? So here what we are learning is that we are using the vectors, mainly the plasmid vectors, we are using the bacteriophage vectors in order to transfer the foreign DNA or the gene of interest into the bacterial cell or into the host cells, right? The bacteria, the viruses, they are able to transfer those genes or the DNA into the host cells. Not only they are able to transfer those DNA or the gene of interest into the host cells, but they are also able to multiply it within the body of the host. How? Because they are able to use the host protein synthesis machinery, so that along with the host protein synthesis, the segment of foreign DNA within the body of the host will also replicate, will also multiply. These are the property, okay, of the vector. So once the vectors will be able to introduce the foreign DNA into the body of the host, into the bacterial cells, those bacterial DNA will be able to use the host DNA replication enzyme, okay, the host protein synthesizing enzyme is their own, and as a result, along with the host protein synthesis process, along with the host DNA replication process, the bacterial DNA, or we can say the foreign DNA within the body of the host, will also be able to replicate, will be able to multiply, will be able to synthesize proteins. Clear? Not only that, but they will also able to change the body of the host also. Okay, so in whatever way they want to change the body of the host, they will be able to change it. So there we have got few examples of the vector which are being used for the transfer of DNA or the foreign gene having the gene of interest into the host, into the plant cell or into the animal cells. So first one is Acrobacterium tumefaciens, which used to cause a crown gall disease in several dicot plants. Okay, so this Acrobacterium tumefaciens, they deliver a piece of DNA called the tDNA, which will transfer a normal plant cell into a tumor causing cells. Okay, and these tumor cells will be able to produce chemicals which is required by the pathogens. Clear? So now the tumor causing cells of this Acrobacterium tumefaciens have the tumor inducing plasmid called the Ti plasmid. That means this Ti plasmid is responsible for inducing the normal cell to develop the cancerous cells. But by the help of this recombinant technology, we can say, okay, this tumor causing gene or the cancer inducing plasmid Ti plasmid has been modified. Okay, this Ti plasmid has been modified, and now it is being used as a cloning vector for the transfer of DNA into the body of the host. That means this Ti plasmid has become now non-pathogenic. It is not able to induce the cancer forming cells in case of the plant cells. It has been modified, it has become non-pathogenic, and now it is being used as the vector to transfer the foreign DNA or the gene having a let's say for DNA having a gene of interest into the plant cells. Clear? Number 2 is retroviruses, which are mainly used to transfer the foreign DNA into the animal cells. Now these retroviruses are also responsible for causing cancerous cells in the animal cells, but the segment of DNA in these retroviruses which are responsible for inducing the cancerous cell formation has been removed. It has been modified. Now these modified retroviruses are not able to induce the formation of cancerous cells in the host cells, and now it is being used is an agent or vector to deliver the gene of interest or the foreign DNA into the animal cell. So kindly remember the two vectors which are being used for the transfer of foreign DNA or the DNA having the gene of interest into the plant cell and animal cell. First one is Acrobacterium tumefaciens and number 2 is retroviruses, and they can be used as a vector only after being modified. Otherwise they are highly pathogenic. Now we are saying that after the formation of recombinant DNA, they are being introduced into the E. coli cell or into the bacterial cells. If you remember, try to recall the formation of recombinant DNA. After taking the foreign DNA, the vector DNA, we are cutting it, we are ligating it, we are forming the recombinant DNA, and after their formation we are putting them into the E. coli cells, right? But the E. coli cells or the bacterial cells, they cannot easily take up those recombinant DNA. They have to make competent in order to receive those recombinant DNA. So now how those E. coli cells or the bacterial cells will be made competent, to take up those recombinant DNA to undergo transformation, we will now learn that. So competence of host cells, the transformation process. Okay, so since DNA is a hydrophilic molecule, it cannot easily pass through the hydrophobic bilayer of cell membrane, and to introduce the plasmid DNA into the bacterial cells, these bacterial cells need to be made competent in order to take up those recombinant DNA. So how they are made competent? So these bacterial cells will be treated with the divalent cation like calcium ion. The calcium ion will make a porous or will make a multiple pore in the body of the bacterial cells or the E. coli cells, and from these pores the recombinant DNA will enter the body of the bacteria or E. coli cells. So now the bacteria with the recombinant DNA will be placed or will be incubated on ice. Then it will be given a heat shock treatment. They will be placed on heat, or they will be exposed to a temperature of about 42 degrees Celsius, and again they will be placed on the ice. Now by this way the host cells or the E. coli cells will be made competent in order to receive the recombinant DNA or to undergo transformation. If you remember, we have already learned about the processes by which we can select the transformed and the non-transformed cells. If you remember the color test, blue-white test, right, and the insertional inactivation test. So those tests can be done in order to just segregate the recombinant cell and the non-recombinant cells. Clear? Okay, now we will learn about the processes of recombinant DNA technology, because there are several steps which are involved in the recombinant DNA technology. Okay, we have got multiple steps, multiple processes which are being involved, which are being used in the process of our DNA technology. The first step is isolation of genetic material, that is DNA. So we have to extract the DNA from the host or from the bacteria. So we have to take up the DNA having the gene of interest from the bacterial cell or from any host cells. For example, gene of interest means having the property of antibiotic resistance. That can be one of the example. Clear? So those DNA segments having the gene of interest needs to be extracted from the body of the host or from the bacterial cell. Second step is separation of isolated DNA fragment. So those DNA fragment which has been isolated now needs to be separated. Okay, they have to be isolated. Number 3, cutting the segment of isolated DNA at specific sites by the restriction enzyme. So after the DNA has been isolated, they needs to be cut. Okay, and this cutting of DNA is done by the help of molecular seizure, that is restriction endonuclease. Number 4 is the amplification of gene of interest by using PCR. Amplification means to amplify, to multiply. A single piece of DNA needs to be multiplied, needs to be amplified to produce multiple copies. Okay, so fourth one is amplification of genes by using the method or by the process called PCR. PCR means polymerase chain reaction. Then after that, there will be insertion and ligation of DNA segment into the vector, and there will be formation of recombinant DNA. This insertion and ligation into the vector is possible by the help of the molecular glue, that is DNA ligase. Okay, so after that that will be formation of recombinant DNA, and next step is insertion of recombinant DNA into the host cells, where we have to see the transformation process. Okay, the next one is we have to select the transform cells by the different techniques which we have learned already. So seventh one is selection of transformed cells, and eighth one is culturing the transformed host cells to obtain clone, that is cloning of recombinant DNA, followed by the last step, which is culturing transformed host cells to obtain desired product at the industrial level. So these are the different steps which are being used in the process of recombinant DNA technology. Clear? So we have to go through each and step in detail. Clear? We have to go through the process of PCR also in detail. Then we have to go to the second step, which is important one, separation of isolated DNA fragment, where we have to learn about gel electrophoresis. So each and every step we will learn in detail. So today we will just learn about the first step, that is isolation of genetic material, DNA. How DNA can be extracted, can be isolated from the host cells or from the bacterial cells. So first one, isolation of genetic material, DNA. So what do you mean by isolation of DNA? We are saying that DNA needs to be isolated. So isolation is the process of obtaining the genomic DNA or the plasmid vector DNA in pure form from the host cells or from the bacterial cells. One more time saying that isolation is the process of obtaining the genomic DNA or the plasmid vector DNA in its pure form from the bacterial DNA or from the host cells. And it has got different steps. We have to go through the first step, second step, and the third step. So we will go through each and every step. So in the first step, we have to isolate. We have to obtain or extract the DNA by breaking up the cell wall or the cell membrane of the host cells. Clear? So digestion of the cell wall or the cell membrane is brought by the help of different enzymes. Suppose we are obtaining the DNA from the bacterial cell, then the bacterial cell needs to be treated with the enzyme lysozyme. If we are extracting the DNA from the plant cell, then the plant cell need to be treated with the enzyme cellulase, because plant cell wall contains cellulose. So cellulase will digest the cellulose. If we are trying to obtain the DNA from the fungus, then the fungal cell wall needs to be treated with the enzyme chitinase. So by this way, the DNA will be extracted from the host cells, but the released DNA will be along with macromolecules, okay, like RNA, protein, and lipids. So even in case of eukaryotes, the extracted DNA will be along with histones, the basic proteins which are being used for the packaging of DNA. Okay, so since the extracted DNA will be not in pure form, because we need DNA in pure form, free from macromolecule, free from RNA, protein, lipids. So therefore in the second step, in order to remove those macromolecules, we have to use the enzymes. For removal of RNA, it has to be treated with the enzyme ribonuclease or RNase, which will digest the RNA. So now RNA has been removed from the DNA. If it contains protein, then it has to be treated with the enzyme protease. So protease will digest the protein. So now by this way, RNA, protein will be removed from the DNA. And for other macromolecules to be removed, it has to be treated with the appropriate processes. Clear? So now DNA will be free from macromolecules. Now we just have to extract the DNA in the pure form. So in the third step, in order to just extract the DNA in its pure form, we have to put chilled ethanol. When we used to put the chilled ethanol, DNA will precipitate, and this can be seen as a collection of fine thread in the suspension, and this can be removed by spooling. Okay, so by this way, by this process, we will be able to extract the DNA in its pure form, free from macromolecules. So today we have just gone through the first step, that is isolation of genetic material, that is DNA, from the host cells or from bacterial cell. The other steps we will go in detail in the next class. Clear? So this much for today. See you in next class. Thank you.